Good afternoon, everyone. Real quick, we'll go through our reminders and then we'll get into the actual lecture as always. One thing I did want to mention, just really quick off the top of my head, because I've gotten a couple of emails about this. If you didn't take the test online and you now have a grade in that slot, that's the grade that I've gotten back from the Scantron people. So don't freak out too much. That does not include your extra credit though. So just keep that in mind. It'll still take me another probably couple days. I'm still waiting on SAS and the ungraded, like the ones that they send back that weren't graded properly. I have to go through and hand grade those. So give me a couple more days. Hopefully by the beginning of next week, I should have those up. Although that may be a little bit difficult depending on how crazy my weekend is. Along those lines, there is a slight chance that I might have to leave a little bit early on Friday to head up to Georgia. So wasn't exactly something I want to do, but it's kind of stuff I have to do with some family things that's going on. So there's a slight chance that I may have lecture online on Friday, but depending on how things go today, we'll see. So just keep that in the back of your mind that you might see an announcement coming from me that, hey, class is only online on Friday, okay? But more than likely that won't be the case. Also, remember that quiz 8 is due on Sunday. I will be at a football game all day Saturday in Athens. So if you have issues, you should probably contact me either before that or after that, because I will not be paying attention to my phone. Also, don't forget you do have that second connected with assignment or connected with biology assignment due. I've already seen where somebody's done it and they did it fairly well so far. So, and they found some really cool stuff. So it's really not that hard. It takes maybe 30 minutes out of your day to just go over there, walk the trail real quick, get done what you need, and then head on back. So just let me know if you have any questions about that. And keep in mind too that there is an extra credit opportunity coming up for this following exam that's very similar to the one that you've already done. I'm not sure if I've already posted it or not, but if it's available, it's really easy. You just literally go through and very similar to last time, you're going to use iNaturalist to observe a certain number of animals, plants, whatever, except for this time you're going to have very specific requirements of how many birds, how many reptiles, that kind of thing. So it's a little bit more work, but it's worth it, I promise. You usually see some really cool stuff. If you have any questions, just shoot me an email, okay? Any quick questions before we get started? Awesome. All right. So today we're going to be kind of finishing up what we started off with yesterday, or we're finishing up with yesterday, Monday, whatever day it was. So we'll be jumping back into mutations, which is any alteration of the genetic code of an animal, right? So anytime you alter the DNA, this is what you end up with. So remember that there are a couple different versions of this. You can have a point mutation, which is where a single nucleotide in the DNA gets swapped, or you can have a frame shift mutation, which is where you either have an insertion or a deletion, where it basically moves that whole framework of how you read those codons over or backwards, and thus it causes completely different amino acid chains to form and thus different proteins. And keep in mind too, with these different things, they can be both silent or they can be expressed. So silent in the sense that you may have a slight change in the DNA, but where that DNA was changed, it doesn't code for a different amino acid, so you don't actually see that difference. So what exactly do these cause these specific mutations to occur? And they can occur spontaneously from errors in the DNA replication. So over time, your DNA is just going to get replicated over and over and over and over and over again. And you will build up some slight mutations that usually don't really mean anything, but they are present in your DNA. Others are primarily caused by things like mutagens, external agents that change DNA structure, things like chemicals. So for instance, when you're smoking, one of the reasons why you have such deleterious effects from it is this carcinogen in smoking, or when you smoke, are actively acting on your DNA and changing it slightly. And also occur through things like radiation. So anything above alpha or beta radiation is pretty nasty. And then things like x-rays, which is one of the reasons why if you go and have an x-ray done, usually the people that are administering it have some sort of lead-lined apron or something like that to protect themselves. And you're only getting hit with it once or twice over, say, a 20-year lifetime. So it's not as deleterious for you, but the person that's going to be sitting there and getting exposed over and over and over again as they're running these things, that's usually why they're the ones wearing all the protective stuff. Now, mutations are not usually harmful. In fact, most of the time, they're those silent mutations like we mentioned, where they're not going to really affect things too much. Most mutations specifically are neutral, and many are even beneficial. So for example, mutations can create genetic variation needed for evolution, right? If you, for instance, somehow manifest a mutation of a specific gene that changes the color of your hair, maybe that allows you to better adapt and find mates easier or something like that. You never know. It can also, and because of this, it can help increase your individual's reproductive success, which we call fitness. So when you hear survival of the fitness, it doesn't mean those that can survive and thrive. It means those that reproduce more. Now, viruses can also do some really cool, funky stuff where they can bring in new genes into cells. So if a cell receives new genes, it can produce a new set of proteins. And so when a cell is infected by a virus, it often receives these new genes from that virus, so that way the virus can replicate. However, if you kill off the virus and kill off those cells, you may still have these leftover batches of genetic material from that virus still in some of those cells. And so it can leave kind of like a genetic signature of you were probably infected with this pathogen, or somebody long down, maybe 50 to 100 years ago that was in your family line was infected with a particular pathogen. It's kind of neat. Now, keep in mind too that viruses are smaller and simpler than cells. And these viruses have very simple structures that lack many of the characteristics of cells. It's again one of the reasons why they're not considered true life. And as a result of this virus does not have a nucleus or organelles or ribosomes, a cellular membrane or cytoplasm. They're pretty much just a batch of DNA that goes in and hijacks the cell and uses the cell to manufacture more of the virus itself. So, but like bacterial cells or human cells, these viruses are going to use nucleic acids to store this genetic information. Now, all viruses contain either DNA or RNA, but never both. And each virus has its own set of genes in its own genome that basically have to infiltrate into the host cell and replicate over and over and over again. Now, these viruses have specialized proteins that are then going to surround these genomes, so that way they can protect themselves as they're moving around the host's body, as well as going in and out of a host. So for instance, in each virus, you have a protein coat. That's the shape of the coat determines the virus's overall form and dictates which cells the virus can infect. And so you've probably seen this example. I think it's probably the classic example this was shown off. If you've ever seen the movie or the show Jimmy Neutron, they do a whole episode with these guys. But that's not the only way and shape that a virus can be shaped. And what's really cool is all of these specialized proteins have specific forms that allow it to basically act like the face hugger from the Alien franchise, where it's going to go on, find a cell, inject its genetic material in it, and then take it over. But then there's kind of more boring looking ones like the herpes virus, which is just a spherical shape. Ultimately, these viruses are going to hijack the transcription and translation of cells. So basically what happens is it puts its gene into that cell then starts to transcribe it into RNA, or if it's already RNA, it gets automatically added to the ribosome itself. And then those ribosomes are going to translate those proteins needed by the virus into whatever form it's needed. Ultimately, though, in order for a virus to exist and produce these proteins, it has to infect the host. And thus, again, this is why it's considered not true life, because it's not living on its own. It has to have that host in order for these things to happen. Now, it's pretty cool how these viruses actually attach to cells. Basically, what will happen is the viral proteins are going to grab onto the proteins of the host cell. The virus is going to inject their genes into that cell. This is usually in the form of either DNA or RNA. And once that cell begins to synthesize that viral nucleic acids, it's then going to start producing those proteins. These cells are going to then produce new viruses. So all those viral nucleic acids and proteins are then going to be assembled into completed viruses. And ultimately, they're going to leave the cell. They can do this in two different ways. They can either just be packaged and sent out like you would send out, say, neurotransmitters in a neuron, that kind of thing. Or they'll burst out of the cell and destroy it when they overwhelm it. Ultimately, viruses can infect all different types of cells. There's certain plant viruses that only bind to plant cells, phage viruses, which are only designed to work on bacterial cells, as well as animal viruses that only bind to specific animal cells. And this can be on a very large level or a very micro level. So you could be talking about things that, say, SARS coronavirus, right? Like infects a wide variety of mammals, birds, bats, tigers, pretty much all large like primates, bottlenose dolphins, us. However, there may be viruses out there that only specifically infect humans. Say, for instance, some of the very specialized sexually transmitted diseases, such as the I think it's herpes simplex type 2, which is the one that occurs on your mouth, that only occurs in humans from what we can tell. So it just kind of depends on the situation and where these viruses evolved, target specific hosts. Ultimately, these phages can kill bacterial cells, and this is called a lytic infection. This is when that virus is going to enter the bacterium. It's going to immediately replicate and cause the host cell to burst, which is going to release a flood of those new viruses. This is the primary way in which viruses are going to actually like take over a host, replicate itself, and then release itself out into the world. But they can also hang out. So phages can use this ability to hide inside of the bacterial cells and just kind of wait around and just, till when there's a perfect environmental opportunity for them to take over. So for instance, this is called the lysogenic infection. These viral genes are replicated along with bacterial genes. The cell lives and reproduces as usual. However, when the cell is under stress or damage, that virus is going to then switch the lytic life cycle and start massively producing those proteins and actually building itself the little virus particles like we expect. And as a result, some of these viruses can linger for years. For example, HIV often remains latent, which does not immediately induce disease symptoms for upwards of 10 years sometimes. However, there's something that triggers that initial response that then causes the virus to become active and start doing damage to the immune system. So HIV has an RNA genome. It must produce a DNA copy before incorporating its genes into the host's DNA. So that's one of the reasons why this latent infection often takes a little bit longer for it to materialize. And don't forget too, that viruses can make plants sick too. The color pattern in these plants might look really interesting to us, but they're actually caused by viruses that are infecting and damaging the all the little pigment producing cells in these plants. Whether it be the green pigment that you use is usually associated with chlorophyll that's being damaged here, or that red pigment there that's associated with probably some sort of attraction for insects or birds or what have you. Now, one of the ways we can prevent these viruses from being able to take over different cells is through vaccines. And like we mentioned with the immune system, a vaccine is simply just a way to introduce those antigens, those signals that, hey, there's a pathogen here, to your immune system ahead of time so that way it can build up and start your adaptive immune response immediately. This is usually our most potent weapon against viral diseases, just because oftentimes they're really difficult to respond to given that they're internal inside of a cell instead of external, like a lot of like bacterial infections and that sort of thing. There are some antiviral drugs that do slow viral replication. There's a new one coming to market specifically for COVID in the next 3 months. That's very similar to Tamiflu, which I'm sure you're familiar with like the flu virus. And what these do is they bind to the cells and block them from transcribing and translating their viral particles in the cells. And because viruses mutate rapidly and are so genetically diverse, it's challenging to produce these drugs with long-term effectiveness. As we've probably kind of mentioned in here a couple of times now, the flu vaccine, right? You have to get it every year. That's because the antigens that are found on the outside of the flu virus rapidly mutate constantly. And it's kind of they call it the Red Queen hypothesis, because you're perpetually, constantly evolving and constantly trying to evade the immune system. Kind of like the Red Queen, or Alice always running from the Red Queen in Alice in Wonderland, similar kind of concept. And that's ultimately why we see such massive divergences in how not only the immune systems respond, but how the host or how the pathogen will respond to the host's immune system. Now, there's also something called viroids, which are non-cellular infectious agents. Viroids are circles of RNA, and they do not encode for proteins, but they do use the host cell to replicate. So, for instance, here's a viroid that you're finding in potatoes. The viroid shown here is thought to interfere with the plant's ability to make essential proteins, and that's why you're getting those weird folds inside the potato itself. So it's still harmful, but it's not a true virus per se. And then you have one other thing that can be kind of a complicated aspect of this whole process that can be troublesome. Those are prions. The prions are non-cellular infectious agents. They're basically, again, a non-living form of something that's somehow found a way to replicate itself over and over and over again. But they don't necessarily evolve like a traditional living organism does. So a prion is a normal cellular protein that sometimes adopts in a normal shape. And as a result, these normal prions can switch over to that abnormal shape if they become into contact with abnormal proteins. These prions are responsible for things like mad cow disease. Especially, you find a lot of neurological damage based on these prion diseases. For those of y'all that are hunters, you've probably heard of chronic wasting disease in deer. That's a really nasty one that basically it's a major neurological disease that completely shuts down the deer's ability to eat, and the animal just wastes away and dies. There's also some really nasty ones that pretty much have a history in cannibalism, particularly in the Congo area of Africa, as well as there's a couple of the islands in the Indonesian island chain that are famous for having, I forget the exact name of the disease off the top of my head, but basically as a result of eating either primates or human brain cells that had these damaged proteins, it introduced these prion diseases, which are pretty nasty and are pretty much fatal if you get them. Now, mutations can quickly result in very different and very varied effects, right? So here's just a simple example where you have a single gene called the FOXP2 gene, which provides clues for the origin of language. So just simply by swapping a few of the amino acids of this language gene, you can see the massive differences between humans, primates, and mice. So for instance, each one of these blocks represents a different chain, right? Or a different chain. So you have a block here that separates where mouse have differentiated from primates. Over here, you can see differentiation between even the great apes. So orangutans are different from gorillas and chimpanzees. And you see two changes that are only found in humans, and that may lead us to understand why humans have been able to develop and have true speech patterns, whereas the other large primates have not been able to. All right. Any quick questions before we move on to the next section? Cool. All right. So we're now going to move into chapter 8, which is simply DNA replication, binary fission, mitosis. So in other words, first we talked about how DNA turns into proteins. Well, we now need to know how do we maintain this DNA and keep it around and then be able to pass it on to cells that are coming from, other cells. In other words, how do cells replicate that DNA and then replicate themselves in that process? Now, as we kind of mentioned before, all cells have to divide at some point. So no living organism can reproduce without cell division, right? Even in multicellular organisms, we have to have some level of this to continue to diversify. Remember when we talked about reproduction at the very end of last unit, you went from that single cell zygote and had to turn it into all these different components of the body, right? So ultimately, unicellular organisms are going to divide in order to reproduce because it's the only way they can reproduce. Whereas cell division is going to produce a continuous supply of replacement cells in multicellular organisms. So it kind of has a multifaceted approach. Now, in the sexual life cycle, obviously, eukaryotic cells are divided by mitosis. So basically, once you have a fertilized zygote, everything from that point on is going to be mitosis. So that fetus is going to grow and develop into a mature adult, consisting of countless cells as a direct result of mitosis. However, in order to get to a point where you can actually have those zygote forming in the first place, remember we have to cut down the amount of genetic material. So we only have half coming from the biological mother, half coming from the biological father. And the reason for that is obviously the more excess genetic material you have, the more likely it is to cause issues. So we use a very similar process to mitosis called meiosis, which is where it's going to have an extra additional step that's going to basically allow that cell to cut in half the amount of chromosomes it has, and then move on to the next step, going from a diploid, so 2 sets, to a haploid, so a single set. And this only occurs during reproductive cycles in sexually selected organisms. And ultimately, the products of meiosis are going to only contain half of the DNA as the regular body cells. And so you have to reunite them with either another sperm or another egg cell in order for that DNA to get back up to its ideal optimum form, and then create that zygote that can then undergo mitosis for the rest of its life cycle. Now, like I mentioned, mitosis has a varied amount of roles. And so you can have mitotic cell division that allows an organism to grow and develop, as well as repair tissue and then regenerate lost body parts. Some organisms can reproduce asexually via mitosis. But here you can actually see a really cool example of this. particularly in amphibians. They have really specialized ability to completely regenerate entirely lost limbs, lost tails, all that kind of fun stuff. Now, cell death is a part of life. So apoptosis or cell death is a process that can carve out distinctive structures during your development. For instance, the hair on your head, and pretty much everywhere else, only like the very basal structure of it is actually still living cells. Everything else is just kind of this leftover dead cell for it. Or for example, with coral, only that very, very top thin layer is actually living cells. Everything beneath that is the calcified skeletons of all the other dead corals that were there before it. So you need these specific structures. So here's a quick review question here. So goat's liver cells contain the exact same DNA as its blank cells. Which of these is correct? So a goat liver cell, right? That's a diploid cell. It's just a regular part of the body, right? So is it going to have the same as a haploid sperm cell? Huh. What about the brain cell? Yes, right? Brain cells are more than likely another diploid cell, correct? What about the heart? A cell in the heart. Should it be diploid or haploid? Diploid, right? So in other words, in this case, only B and C are correct. So DNA replication is linked to cell division. So cells have to copy their DNA before they can divide, because you need to have two copies, right? You need to have one in the new cell and one in the old cell. So that's why they have to go through this cellular or this DNA replication process. So the entire genome is replicated so that each new cell gets one complete copy of that DNA strand, right? Now in DNA replication, both strands are completely copied. Now remember that both of them are complementary, right? So if you got an A on one, you're going to get a T on the other and vice versa. And basically you can read them from 5-3 or 3-5, depending on what side you're looking at. Now what's kind of nice about that is given that you know that these are complementary strands, right? That means they're relatively exactly the same. So you can take a half strand from the other and a half strand, the other half strand, and then replicate that complementary strand alongside of it. And now you have two of them perfectly matched to each other. So this DNA replication is what we call semi-conservative, which basically means that cells keep the original parental DNA each time that replication takes place, while producing a new strand at the same time. In other words, you have half of the strand, it's going to then add this daughter strand on half of it. And so that way both sets of cells, or the new cell and the old cell, are going to end up with half of that original DNA. Now, after replication, each DNA molecule is going to have one parental strand in that dark blue and that one daughter strand in the light blue. And again, this is to help minimize, which is that whole semi-conservative thing, right? To help minimize damage and to kind of keep things as consistent as possible from one generation of DNA to the next. Now, enzymes are incredibly responsible for this whole process and are basically the main reasons why you have DNA replication in the first place. So first, you're going to have an enzyme called helicase, which is going to unwind the DNA itself. So you're going to basically take that double helix structure and straighten it out and then break it apart. Then you're going to have the DNA polymerases that are going to come up behind it and start assembling the new nucleotides onto the parental strand and create that new daughter strand. And then finally, you're going to have ligases, which are going to join the short strands and the long strands together and form that new piece of DNA. Ultimately, that complementary base pairing is going to assure that new DNA strands follow that same correct sequence that we want from the original parent. Unsurprisingly, like everything else in life, requires a lot of energy. ATP is required for every chemical reaction, just like, and obviously you're going to need it for DNA replication too. So energy from ATP is going to be used to produce the nucleotides, unwind the DNA, and bind the new strands. So it has a bunch of different roles here. These replication enzymes are going to divide up the work ultimately. So you have this DNA replication becoming simultaneously at multiple spots along the chromosome called the origin of replication. And this replication is going to proceed in both directions at once from each origin until they're ultimately, taking up the entire thing. So it's kind of like, you've ever seen somebody build a ship, right? They're not just working on one section and then moving slowly down the thing. If you've ever seen them, especially when they're laying the hull, they're all working together, building the entire thing at once. Exact same process here. What's really cool though, is we can actually control some of the enzymes as well as temperature, which will act very similarly to some of these enzymes because we can denature the DNA without having to use a specific enzyme to replicate DNA without a cell. This is called PCR, or polymerase chain reaction. Basically, we've taken the Thermus aquaticus, which is a type of bacterium. We've taken their DNA polymerization enzymes. We've extracted it from that cell, and based off of the temperature that we know that it works at, we can use specific temperature regimes over, say, a 2-minute time span to take a strand of DNA and turn it from 1 strand to 2 strands to 4 strands to 8 strands, basically logarithmically increasing every time we do it. And it has a lot of really valuable uses. Now, obviously, if you need to sequence DNA, you're going to need more than a single copy. One, because there could be damage to that copy, and so you might be missing information. But 2, in order for it to really work effectively and sequence a wide swath of the genome of an animal or plant or whatever, you need as much genetic material as possible so you don't miss anything. And there's a lot of ways through supercomputers in the back end to basically merge and figure out exactly where, okay, I have base pairs 1 through 1,000. Here's base pair 750 to 1250. You can merge them in together and know exactly where they're supposed to fit. Then you can also use it for some really valuable things like detecting diseases or the presence of an organism through something called qPCR. qPCR basically is simply where we just bind an extra little particle to that DNA that we're making. It allows us to flash a little hit of light to it. And based off of how strongly we get that reflection back, we can determine how much DNA is present at a specific time period. So if you're looking for diseases in host tissue, you're not sitting there looking for a specific virus. You can just look for the particles of DNA present in that tissue from that virus. It's really cool. Additionally, there's a really useful for this application for this called eDNA, which is where you can literally just go out in the woods and collect soil or collect water out of a river and search for particular animals, particular plants, all that kind of fun stuff, just by detecting the DNA that they left behind. Really useful. All right, so let's do another quick review question here. This image at the right shows a cell of DNA before replication, where you have 2 parental strands. Which of the following best represents the newly created red strands after replication is complete? Either A, where you have a completely separate parental and daughter strands, or B, where you have one of each. B, right? And again, that's that whole semi-conservative approach that we talked about. It's there so that way you're not losing anything by accidentally having mistakes happen during the DNA replication process. Now that you've got this DNA replicated, let's kind of look at the actual breaking apart process and looking at how cells go through now that they've had this DNA, extra DNA that can now separate into 2 distinct cells. So in prokaryotes, they're going to divide primarily through binary fission. So in bacteria and archaea particularly, you have this asexual reproduction that occurs via binary fission, in which they basically replicate their DNA, distribute it, and then distribute it into 2 distinct daughter cells. This process is called mitosis, and we'll go through each individual step here shortly. Ultimately, just know that you start with a single copy of DNA in a single cell. You duplicate that DNA. You then split the cell into 2, creating these little cleavage points. And ultimately, that's going to result in 2 distinct cells with 2 copies, or each with a single copy of the DNA itself. Now, prokaryotes, despite only doing this asexual reproduction, can get new DNA. Obviously, you have mutations, right, that we've talked about. But bacteria and archaea also have what they can take apart, what they call a donor cell, which can directly transfer its genes into a recipient cell. This is usually through something called the sex pilus. Pretty straightforward. And healthy cells can also absorb DNA from dead and dying cells and incorporate it into their genomes as well. Basically, if you have this chromosome fragment floating around freely in the environment, a bacterial cell may choose to just incorporate it automatically into its own genome. This can also take the form of plasmids as well, which are just free-floating, specially designed DNA or a set of like DNA genes that can then just be kind of sent out into the world, and then that plasmid will be uptaken by other bacterial cells, and as a result, directly have this new DNA come into that new population. So question 3 here, why is binary fission an asexual process? Pretty straightforward answer, right? C, right? So it produces 2 genetically identical cells. That's all you need to remember here. Now, eukaryotic DNA is going to wind up extra tightly before cell division. That's why when you normally think about DNA in cells, it's going to kind of look like a loosely packaged kind of clump of DNA. It's still packaged. It's still not like all just a single strand wound out completely, but it's still a lot less easy to work with. And so just prior to mitosis, you're going to need to actually package that. God, that's fucking obnoxious. You need to package that DNA in a more discrete way. It basically, it's like the difference between just carrying around a big ball of yarn versus like having nice discreetly packaged like little sections. It's only Wednesday, right? Damn. All right. So as a result, these eukaryotic chromosomes are going to be highly condensed DNA. And basically what happens is you have a single copy of DNA and a single copy of DNA. You have these centromeres here, which is right down together. And so each one of these chromosomes is going to consist of these 2 identical sister chromatids, one for each copy of that replicated DNA. So remember, sister chromatids, they're identical. They're on the same chromosome together. That will come in handy to know later on. I promise you. Now, these eukaryotic chromosomes are extremely visible during cell division. You can actually dye the cells so you can see this. In fact, during like the regular intro to bio classes that we teach for the major students, we have them do this in person, which is really cool. But you can actually see, hey, for instance, here's like just all that free floating chromosomes here. And then you can actually see right before cellular division the specific chromosomes themselves. Now, mitosis, remember, is going to generate exact cell copies. It's that asexual process, right? So even in eukaryotes, it's still, even though it's not truly replication or reproduction at that point, it's still that clonal process, relatively speaking. These eukaryotes are going to divide into that process called mitosis, and mitosis is one part of the cell life cycle. Now, this isn't a majority of the cell life cycle. In fact, most of the time, the cell is just be existing and functioning as a regular cell. This is just how it gets through the reproductive stage. Now, at the end of mitosis, 2 new daughter cells are going to have the exact same DNA as the parent cell, as well as usually the exact same amounts of things like all the different ribosomes and that kind of fun stuff, all those little cellular organelles. Now, a cell cycle is a repeated series of events. Ultimately, the cell cycle is going to last from the beginning of one cell division until the beginning of the next. And so even though we consider it like a mother cell and a daughter cell, there are 2, we kind of just stop thinking of them as the same thing at some point. It's not like you have an adult that has a child and that has a child. It's each time it's a brand new cell, essentially. So it includes times when the cells are dividing through mitosis, as well as the time when the cell is not dividing, which we call interphase. Inter as in between, that kind of thing. Now, during interphase, that's when the cell is not dividing, and that's when it's going to be carrying out its normal everyday functions unrelated to cell division. So if it's a liver cell, it's going to be producing specific enzymes. If it's a red blood cell, it's going to be out there moving around, oxygen particles, that sort of thing. For instance, as cells express their genes and carry out active transport and perform those life chemicals reactions that you need for a eukaryotic organism to exist. Now, interphase is also going to include what we call the G1 phase. That's when the cell grows and functions normally, and that's when protein synthesis occurs. And if the cell is getting ready to divide, it will then move into the S phase. And if not, it's just going to stay in that G1 phase perpetually for a while. Now, interphase still includes this S phase, but this S phase is where you're actually going to start seeing the replication of DNA. It's going to copy its entire genome, every single chromosome, and now it's committed to divide because there's no reason to have all this extra DNA if you don't need it, right? So there's no going back after this replication begins. It's pretty much an immediately process after that. After that S phase, it's going to enter what we call the B2 phase. This is when the cell is going to start producing specialized proteins that are necessary for mitosis to happen, but it's still going to be able to kind of carry out some of its functions as a cell. And finally, it's going to enter that M phase, which is the mitosis phase, which is where you're actually having that cellular division. This is where you're going to start seeing the division of the nucleus, which is then followed by cytokinesis and the division of itself. Now, mitosis is a series of very specific steps summarized here, and we'll show in more details in the following slides, so bear with me. So first, you're going to start with the prophase, which is where the cells are going to have to condense all that replicated DNA that they have into those very compacted chromosomes that we showed earlier, so that there's 2 copies of the genome that can be separated easily later. Then at the exact same time, you're going to have the formation of what we call spindle structures, which are basically made of the microtubules left over from the internal parts of the cell itself, and are going to be there to basically pull apart the cell, as well as pull out the specific copies of the genome itself. Here specifically, you can see where you have that early prophase and late prophase, as those chromosomes are condensing and those spindle fibers are forming up. You can see it both in this diagram here, as well as actually what this will look like in the cell itself. Ultimately, at the very end of prophase, you're going to have the actual dissolving of that nuclear envelope, and the spindle fibers are going to attach to each chromosome. Prophase is then immediately followed by metaphase, which is where the spindle fibers are going to align the chromosomes in the middle of the cell, where 2 chromatids are still held together. So see, they're all still nicely kind of paired up together. You still have those 2 sister chromatids. They're not quite separated yet. This is ultimately what we call the equator, and that's usually going to be exactly where the chromosomes are going to line up. Think of, if you've ever done anything with geography, you know what an equator is by now. That's the center point of the Earth, right? And this arrangement is going to ensure that each cell will receive exactly 1 copy of each chromosome. Metaphase is then followed by anaphase, which is where the 2 sister chromatids are going to split apart and are going to be pulled apart by the spindle. Now, the spindle is also going to start to stretch the cells out. So you're going to go from like that perfect circular shape to it's going to start stretching on the boundaries of it and kind of get more of an oval, oblong egg shape to it. Specifically in anaphase, you're going to see these spindles separate those chromatids. A little bit easier here in this diagram to see it, where you've actually got these chromatids splitting apart from each other from the 2 chromatids. And that spindle is going to shorten, moving those sister chromatids towards the opposite side of the centrioles, which is going to be ultimately where the new nucleus forms. Now, anaphase is then followed by something called telophase. These are the 2 copies of the DNA are at the opposite ends of the cell, and ultimately it's going to get repackaged into a new nucleus. So this new nucleus is going to form around each one as the cells get ready to split in half. So again, here you can see what actually looks like both on the diagram. The actual cell itself. Basically, the centromere are now pulled all the way to the end of the cell. They've repackaged all those sister chromatids as a single copy in each side of the cell into a new nucleus. And you can see that pretty easily that they're very clearly getting ready to divide. And finally, this results in what they call cytokinesis, which is where you actually get the splitting of the cell. This is where the cytoplasm and the 2 nuclei are distributed into the 2 forming daughter cells when they physically separate. Ultimately, the cytoplasm is going to divide into 2 distinct cells. And basically, you can see where you've got this contractile arrangement that basically the cell membranes kind of merging back together. It's kind of like if you've ever split apart like a little droplet of water, you're just going to end up with 2 smaller drops of water, right? It just kind of does that weird point where it's going to have that tension on either side. This is what we call a cleavage furrow, and you can usually see it very well in animal cells. And it's just simply the indentation that forms at the middle of each cell as it's dividing. Pretty straightforward. Now, the reason you don't see this in plant cells, what's different from a plant cell versus an animal cell? The cell wall, right? So in cytokinesis and plant cells, the cell plate forms between those plant cells where that cytokinesis is happening. And so the cell plate is the beginning of a new cell wall that's going to separate those 2 daughter cells from each other. And ultimately, vessels are going to gather at the middle of the cell to deliver those necessary cellulose and other polysaccharides and proteins so it can actually form that true cellular wall between those 2. Another quick review question here. So in some fungi and slime molds, mitosis may occur without cytokinesis. What would you expect to find in these species? So if you've gone through all of mitosis and you don't go through cytokinesis, what is going to happen? You're going to have a cell with a lot of different copies of nuclei, right? Basically, it's just one giant and large cell with no true separation, with a lot of distinct little nuclei that are all kind of acting together. They're really kind of funky to see. Those slime molds in particular are really fascinating because they, surprising for a very basic organism, are able to like find and do resource allocation better than humans can half the time. Really cool. Ultimately, this cellular cycle is under extremely tight control, where you have a series of chemical checkpoints that are going to regulate it. These checkpoints ensure that all DNA has replicated, the DNA is not damaged, and the chromosomes are lined up and separate properly. Because if at any point any of this goes wrong, you'll have basically a destroyed cell that doesn't function. Essentially two, depending on how bad the error was. And that leads us to what happens when this goes a little bit wonky. Now, cancer cells specifically are cells that have divided out of control. There's some sort of effect, whether it be a prion or some sort of mutagen that's affected the actual genome of that cell and cause that cell to just massively replicate without any real form or function anymore. So if you've ever seen like what a cancer mass looks like, kind of big and globular and lumpy, it just looks like something just kind of, it's like the blob or something like that, where it's just kind of growing out of control. That's why. These cells are going to continue to divide, and when they do not need to, they override those checkpoints and just keep on dividing over and over and over again, which is why they don't maintain that sort of shape or structure. They just kind of keep growing. This is where you're going to produce those actual tumors itself, which is an abnormal mass of tissue. This is where cancerous cells have divided out of control and formed this mass. And those tumors are either benign, which means they're contained and they're not growing out of control, or they're malignant, which is where they can take over other types of cell tissue and start this massive outgrowth in other places. So even though it might be considered benign, it still can be cancerous. So keep that in mind. Ultimately, the only way you're going to get rid of these kinds of cells is you're going to have to remove them. And it's one of the reasons why they have to not only cut away things, but you also have to do those subsequent follow-up things like chemotherapy. And the reason why chemotherapy is as effective as it is, it's targeting very quickly replicating cells. So the reason why it affects, say those cancer cells, is the same reason why it affects hair cells. So the reason why people often shave their heads or just go bald on their own is because that actual cancer, the chemotherapy will target rapidly dividing cells inside the hair follicles and basically stop that from happening. Same thing with a lot of the sickness that's usually associated with chemotherapy, particularly for stomach pains and all that stuff, is because it's killing the rapidly dividing parts of the stomach that are there to help process foods. And so it just doesn't quite work as well as it should. But ultimately, in order to get rid of the cancer, you're going to have some sort of surgical removal of some kind to actually get rid of the mass itself, as well as anti-cancer drugs that are going to keep those cells that weren't completely removed from replicating further. Ultimately, radiation is there to help kill those targeted tumor cells. So like we mentioned with those mutagens, there's a lot of different ways you could potentially run into situations where you're going to have things corrupting your DNA. So just keep a lot of these things in mind. Obviously, obesity, tobacco use, ultraviolet radiation, marijuana use, alcohol use. There's specific viruses that can cause cancer, things like HPV, I believe a couple of others as well. And one of the big things, especially with cancer, is detection time. So if you know you have family history with specific things, because there are certain genes that just tend to code for cancerous cells, keep an eye on that. And ultimately, this is one of the reasons why we'll never truly get rid of cancer. There's no such thing as a cure for cancer. It will never happen. And the reason for that is because it's so highly inconsistent between the individual, the types of cells that are going to become cancerous, all that kind of thing. So it's impossible just to have a single drug that's going to kill every single kind of cancer. You may get lucky and have some things that are more beneficial in particular circumstances, say breast cancer, where there's been billions of dollars spent on research, and they found a couple things that work. But this isn't always the case. And again, chemotherapy is going to be there to help kill those susceptible cells, and it changes the balance of power and allows the healthier cells to be free from that competition get access to nutrients and be able to reproduce again. So that way it's not all taken over by those cancerous cells. There's a lot of different techniques that we are investigating now. So for instance, this adaptive therapy is a new approach, which is where you use declining doses of chemotherapy to let some of the susceptible cells survive to be able to compete with those resistant cells. So that way you're not just killing off a wide swath, all of the cells. You're just only, you may still have some of those cancerous cells still around, but you have the more healthier, normal cells that are already there and present, kind of taking over and acting in that regard. All right. As always, don't forget quiz is due on Sunday. If you have any computer issues, try to get a hold of me sooner rather than later. As well as start working on connected biology too. It's due at the end of the month.